Thank you. I'm Wei Li from Chinese Academy of Sciences from Beijing. First, I would like to thank the organizers for inviting me here. Actually, this is my first time to visit Korea. Last night, I told Jian Wang in the dinner that it's really a beautiful place. Just let me remind my hometown China, a lot of mountains and even a lot of rivers in the city. It's a very beautiful place. So today I just changed my title a little bit because today I will mainly focus on the embryonic stem cells. I will mainly present the works collaborated with Qi Zhou, the president of the CSSCR, who was initially invited, but he couldn't come due to some reasons. So we know the stem cells is a really amazing cell type. They have many applications. So generally, these applications can be divided into two parts. The first, they are widely used in basic research. For example, we use stem cells to mimic the development and disease. And they are also used as biological tools. For example, we combine the embryonic stem cell technology and the homologous recombination to do gene targeting. And the other application is regenerative medicine. We differentiate the cells, stem cells, to functional cells and use the functional cells to do cell replacement therapy. So today I also follow the two lines to introduce our work on both the basic research and the translational work of embryonic stem cells. So the embryonic stem cells have a very amazing and unique feature called the pluripotency. The pluripotent stem cells can actually form all the cell types in our body, including the germ cells and even the actual embryonic cell types. So there are many sources of pluripotent stem cells, including the embryonic stem cells and induced pluripotent stem cells, and even other cell types like the germinal stem cells. So our goal, we have studied embryonic stem cells for a very long time. We try to understand the regulation of the pluripotency of the stem cells and how they are established, how is the pluripotency established, and how is it regulated, and why is the pluripotency of stem cells from different species just exhibit different features. We mainly use the functional assays to assess the pluripotency of stem cells from different species. For the functional assays, there are many types of assays. The most stringent assay is the tetraploid complementation. This approach is just like the somatic nuclear transfer, like the cloning. By this approach, the whole animal is produced from the stem cells. So many years ago, we published a paper. We proved that the mouse induced pluripotent stem cells can produce a viable mice through the tetraploid complementation. And more recently, we have tried to produce rats from the rat embryonic stem cells, and we have shown that we just found that the very early passages of the rat embryonic stem cells can produce full N rats, but the late passages, even after passage 10, the stem cells lose the ability to form the full N rat, to produce the animals. So mainly because the embryonic stem cells in the culture experienced a global demethylation, and they just lose the ability to produce animals. This paper has been rejected by many journals, but recently, last week, two papers published in Nature, they showed that actually the mouse embryonic stem cells also experience the same status. They experience the disposal to genome-wide demethylation and lose the ability to form the to produce the animals by the tetraploid complementation. For the primates, it's really difficult to assess the pluripotency by in vivo assays. So we are trying to establish an in vitro assay to study the differentiation potential of the pluripotent stem cells. And last year we published a paper in Cell Stem Cell. We tried to generate gametes, the sperms from embryonic stem cells. And this is a published protocol that by many procedures we can differentiate the embryonic mouse embryonic stem cells into haploid cells, just mimicking the round sperm. Also, work from Sato and Gyato's lab also demonstrated the embryonic stem cells can differentiate into functional oocyte in vitro. So I think these works have just showed that actually we can constitute the whole life cycle in the dish in vitro. So I think these are very useful tools to study the epigenetics and the cell cycle regulation of in biology. Today I mainly will introduce a work related about the haploid stem cells. We know that in mammals, the whole life cycle is restricted to diploid status. A haploid sperm and a haploid oocyte are fused to form a diploid embryo, and only the diploid embryo can form the organism. So we are trying to generate an in vitro model of haploid stem cells. In mammals, the haploid only exists in highly differentiated cell types like the gametes. The gametes are very hard to culture and to difficult to be derived. So we are trying to derive the haploid stem cells, and we have this is a general this is a simple procedure to derive the haploid stem cells. We can derive both the haploid stem cells from a sperm or from the oocyte. For the sperm-derived androgenetic, we call it the ES cells, the androgenetic haploid stem cells. They are derived by transferring the sperm into a enucleated oocyte for the construction of a haploid embryo, and then we can derive haploid embryonic stem cells from these haploid embryos. And we also demonstrate these sperm-derived androgenetic haploid embryonic stem cells. These ES cells can produce the mice by substituting for sperm by injection into the oocyte. We also found that the oocyte-derived parthenogenetic stem cells, the PH haploid embryonic stem cells, can also produce mice by substituting for the oocyte genome. So this is really amazing that these haploid stem cells not only mimic the ploidy feature of the gametes, they also maintain some gamete-like feature, epigenetic features. Only the sperm-derived AHES cells can function as a sperm, and the oocyte PHES cells can take place of the oocyte genome. We mainly use these haploid stem cells to study the basic questions for reproduction, and also for some evolutionary studies. we know that I think a paper published in Science around 20 years ago, the scientists just compared the genomes of the monkeys and chimpanzees to the humans, and a very striking finding. They just showed the genomes of the chimpanzees and the monkeys are highly similar to the human genomes. So they proposed a theory. The evolution of the species not only driven by the genome sequence variations, but also by the gene regulation theories. So the hybrid cells and even the hybrid animals actually are widely applied to study the gene regulations between the species, because we can place two divergent genomes into the same cell nucleus, and they just face the same nuclear environment. But for evolutionarily distant related species, for example, the mouse and the rat, they just cannot produce hybrid cells. So by fusing the haploid stem cells from different species, we can actually create the allodiploid stem cells. This paper has published. We fuse the mouse-derived androgenetic or haploid ES cells with rat-derived PHES cells to create allodiploid embryonic stem cells. Actually, they can maintain a stable diploid status, but they also maintain both a set of mouse genomes and a set of rat genomes. And these cells, they also have the features of the embryonic stem cells. They are pluripotent, and they can differentiate into all three germ layers and even the early stage germ cells. And they also exhibit unique gene expression patterns, including the additive expression, high parent and low parent expression. So we can use these unique allodiploid stem cells to study the gene regulation of different species and to compare why how they behave differently and how they maintain this. Here is just an example that we use allodiploid stem cell to screen for genes related to species-specific phenotypes. We know that the embryonic stem cells from different species, they just exhibit different features like different morphologies, and they rely on different signal pathways to maintain the pluripotency. So we just compare the mouse and the rat gene expression in these allodiploid stem cells, just like a molecular sieve to screen for the candidate related to species-specific phenotypes. And we just found that actually the Wnt signal pathway, which has also been reported by the Austin Smith lab, that has been differentially expressed between the mouse and the rat. And also we found that the TGF-beta signal pathway has been differentially regulated in the between the mouse and rat embryonic stem cells. So actually we have proved that these allodiploid stem cells could be a useful tool to study the species-specific phenotypes. Here I would like to introduce some unpublished results that we use haploid stem cells to study the genomic imprinting. Genomic imprinting is epigenetic phenomenon in mammals that lead to the parental specific expression of imprinted genes. The imprinted genes are only transcribed from the one allele. For example, the paternally imprinted genes are only transcribed from the maternal allele, and the maternally methylated imprinting genes are only transcribed from the paternal allele. So these regulations just make the paternal genome and maternal genome different by functions. So both the paternal genome and the maternal genome are required for normal development. So why is the genomic imprinting is evolved? The evolution arrangement, many scientists study this question, and I think it's very important theory has proposed by David Hack. They just proposed that the genomic imprinting is evolved by due to the parental conflict, or also called the kinship theory, because the parents have different requirements for the offspring development. The fathers just want the offspring to grow large, to be more strong, because only this can adapt to the evolution in the nature. But the mothers, they just keep the offspring small, because they can spread their resources to more offsprings. So the parental conflict is the major theory, and the kinship theory is the major theory for the evolution of genomic imprinting. And we just we just established an experimental approach to study the genomic imprinting. We know that genomic imprinting is mainly regulated by the cis regulatory elements, like imprinting control regions. These regions are differentially methylated by the genome. We found that the haploid stem cells actually are deposed to genome-wide demethylation, and the PHES cells just have a similar expression methylation level to the primordial germ cells. We know that in our body, only the PGCs have maintains a very, very low methylation level, but the haploid stem cells actually also maintain a hypomethylated genome. And all the elements, including the promoters, exons, introns, and the transposons, and even the imprinting control regions are disposed to demethylation. For example, the H19 and the IgDMR of the DLK1, DL03 region. So we just mimic the hypermethylation of the genome by deleting the imprinting control regions. So this is a general description of the approach. We use the haploid stem cells to study the cis regulatory elements in animal level. We just use the CRISPR-Cas9 and paired sgRNAs to transfect the haploid stem cells and delete the cis-regulated elements like the imprinting control regions, and then we construct embryos from these CRE-deleted haploid embryonic stem cells, and we can directly observe the phenotypes of the deleted CREs in the mouse. Here I quickly go through the results. We showed that we found that by deletion of 7 DMRs, the differentially methylated regions in the HES cells can enable the generation of live births of full-term by paternal mice. We also found that by deletion 3 DMRs, we know that in mammals, the paternally methylated regions are mainly related to 3 DMRs. We showed that by deletion, all of these DMRs, we can generate actually novel development of bimaternal mice. So this is just a procedure. We can use the oocyte from a mouse and derive pathogenetic haploid stem cells and do DMR modification and injection the PHES cells into the oocyte of another female mice and to produce bimaternal mice. And these mice can just develop novel very interesting finding that we found that by deleting different imprinting control regions, actually the mouse showed different phenotypes. But basically, we found that the paternal methylated regions just controls the paternal expressed genes are just promote the growth of the offspring, while the maternal expressed imprinting genes just restrict the imprinting genes. So these experimental findings are just support the kinship theory of the evolution of the genomic imprinting. And a very interesting finding is that the paternal mice have a prolonged lifespan. So that means if they didn't express the Ras-Shirfan genes, they can maintain a longer lifespan. So this means that the Ras-Shirfan, the genomic imprinting, is actually related to the growth and even the aging of the mice. But how to why the genomic imprinting is related to lifespan? We thought that because in nature, the major function of the animals just try to transmit their genome to next generation. For humans, we try to prolong our lifespan. We want to live all the times, but in nature, actually, after reproduction, actually they don't need to maintain their body. The best way for evolution is just to transmit the genome to the younger generations. So I think this is a very interesting question that what's the side effect of a prolonged lifespan? Actually, we found that in the bibarnatona mice, even they can have a major lifespan, but they also have altered cholesterol metabolism. So I think this is a very interesting question about the evolution arranging of the aging. Why we should age and how this is regulatory mechanisms are evolved by the nature. So what's the side effect if we use some approaches to prolong the aging? I think this is very interesting questions. Probably the kinship theory also give us some insight into this question. So finally, I just quickly go through our study on the translational part, because we don't know the result yet. So we have generated a stem cell bank in China. It's the first stem cell bank for the clinical applications. All these stem cells and stem cell derived functional cells are clinical grade. And we also published the paper in Stem Cell Reports to describe how to produce the clinical grade stem cells, which combines the guidelines by the ISSRs and the Chinese regulations to derive this stem cell. We have used the clinical grade stem cell, embryonic stem cells to generate dopamine neuron progenitor cells, and we try to use the stem cells to cure the Parkinson's disease, and we tested the functions of these neurons in the monkey brains. So we have traced the study by 5 years, more than 5 years, 6 years. 6 years ago, we transplanted the embryonic stem cells derived dopamine neurons into the monkey brains, and we just studied the monkey brains last year, and no tumors was found, and actually some transplanted monkeys showed a recovery of the disease phenotype. So this is a video for the, sorry, okay. This video just showed the recovery of the disease phenotypes by DA neuron transplantation. We found that the DA neurons can actually integrate into the monkey brain, and they don't produce any tumor or other side effect. So now we have just launched a clinical study to use the human embryonic stem cell derived DA neurons for Parkinson's disease treatment. And this is actually the first clinical study in China to use the stem cells to treat Parkinson's disease. So finally, I would like to thank all my collaborators, including Dr. Qi Zhou and Hu Baoyang and Wang Xiujie from the Chinese Academy of Sciences, and other professors, including Zhao Xiaoyang from Southern Medical School, and Ji Weizhi from Kunming Primary Center. And all the students have contributed a lot to our studies. So thank you.